All right, how do we use ATP? How do we couple favorable and unfavorable reactions in living things? And how do we do chemical reactions in metabolism? The answer is enzymes. Enzymes are tremendously important in biology, in all of the chemical reactions of biology, including the reactions of metabolism. So we're going to have to describe and understand how enzymes work. To understand how enzymes work, we have to start out by pointing out that most organic reactions are slow. A really good example of a chemical reaction is probably right in front of you right now. Somewhere around you, even though you're looking at a computer, is probably a piece of paper. Maybe you're taking notes on a piece of paper, or you certainly have paper around you sometimes during your education. Paper is always undergoing a chemical reaction. Paper is mostly made of glucose. Glucose polymerized together in a polymer called cellulose. And glucose, and therefore cellulose, is always being broken down into carbon dioxide and water. But it's going very, very slowly. If you take that piece of paper that's in front of you and you look at it in a few hours or a few days, you won't notice that it's any different. But what if you come back and look at it in a few decades or in a few hundred years, or like this piece of papyrus paper, in a few thousand years? This paper has been oxidizing for thousands of years, and it's literally falling apart because the paper is literally turning into carbon dioxide and water. But it's not going very fast. It's going very, very slowly. Well, the reactions of organic chemistry are mostly slow, and organic chemistry is the kind of chemistry that happens in cells. But cells don't have to wait around for reactions to go that slowly because they have enzymes. The reason that reactions are slow is that they have big activation energy. So we talked about a favorable reaction being like rolling a rock down a hill. But a reaction is not just a change in energy from the reactants to the products. There's a period between them. And so the form between them, called the transition state, that form can be in higher energy than either the reactants or the products. If there weren't a hill here, all of the reactants would already be products. We wouldn't observe the chemical reaction because all we would observe is the products. But in a slow reaction, there's a hill that you have to get the rock over before it will finally tumble to the end. So paper oxidizing is favorable. Paper will eventually spontaneously oxidize and become carbon dioxide and water. But why is it not oxidizing fast? Because it has this energy of activation barrier, or activation energy barrier, that it has to get over. You can help that. If you take a piece of paper, you can put some energy in, like light a match under it. And what will happen? Well, then the whole paper will combust, completely oxidizing, becoming carbon dioxide and water really quickly. That happened because you put energy in. But and the burning paper will end up putting more energy, more total energy out. Your burning match put a little bit of energy in to get you to the top of this hill, but then the whole reaction put more energy out. And you could, warm your house by burning paper. Don't try that though, please. That slow reaction is slow because of how high this energy of activation barrier is. Enzymes are catalysts. Catalysts make reactions fast. How do they make reactions fast? They decrease the height of the energy activation, the activation energy hill. I keep saying energy of activation because that's one way that this is termed, but activation energy is another. When you use an enzyme, the energy of activation, or the activation energy, is shorter, smaller, and so the reaction doesn't have to wait for as long to get enough energy to actually happen. The reaction goes a lot faster. The activation energy is shrunk by having an enzyme. Okay, how in the world does an enzyme do that? An enzyme is a protein. Remember in the section on the molecules of life that we talked about proteins as large complicated molecules. We said that there are thousands of different proteins in a human being. In fact, you have genes that encode from your DNA in a way that we'll see later in the course, tens of thousands of different proteins. And most of those proteins, well over half of them, the majority of them, are enzymes. Here on this slide we see an example of a protein. This three-dimensional shape is the shape of that molecule. All these little bumps are individual atoms, and there are actually thousands of atoms. This is a relatively small protein, but there are thousands of atoms in this molecule. This spaghetti stuff and these coils and strands are the structural elements of the protein. If we just trace the amino acid chain through the whole protein, we would trace through all of these lines. This protein folds into its three-dimensional shape, and in that three-dimensional shape there's this surface. This surface, that is this crevice here, is the special surface that makes catalysis happen. That's where the reactants, called substrates, the reactants of an enzyme catalyzed reaction, sit and get positioned with each other so that the energy of activation, the activation energy barrier, becomes lower. It's the shape and configuration of the enzyme, that special surface on the enzyme, that makes catalysis happen, that makes the reactions go faster. That's what enzymes do. They make reactions go faster. But what does that have to do with energy? Well, some enzymes, not all, in fact, not even a majority, just a few, but some enzymes make unfavorable reactions go by coupling them to favorable reactions. Some enzymes, not the majority, but some, catalyze more than one reaction at a time, two or sometimes even more than two reactions. A favorable reaction and an unfavorable reaction, that couples them. And coupling the reactions allows the unfavorable thing to happen because the favorable thing is happening. For instance, we can have an enzyme that is catalyzing the reaction, putting A and B together to make a new compound, AB. Now that's a synthesis reaction, and it might be energetically unfavorable. Let's say that it is unfavorable. But you need to make AB because maybe AB is a growing protein or a growing piece of DNA or something. And you need to put the pieces together. How do you do something that you have to do that's unfavorable? How do you get the 5 kilogram mass, 5 kilogram weight, to rise? The answer is you couple it to something that's favorable. So on this same enzyme, not only do we catalyze A and B becoming AB, but we also catalyze ATP getting hydrolyzed to ADP and phosphate. That is favorable, and this is unfavorable. Together, the whole thing will happen, so that the unfavorable thing happens because you're spending some ATP money, some ATP energy. You're dropping a heavy weight in order to lift the lighter weight. The enzyme is like the pulley. It's the thing that couples them together.